everyone, I'm Emma. Today I'm going to take you through the entire Paper 2 Biology for GCSE Combined Science. If you study higher tier, those parts are labelled like this, and if you study foundation, you'll know just to skip those. Of course, this is just the content. Make sure you practice lots of questions. If you watch my individual videos, you'll have quick questions at the end, and I've got other videos like maths and biology to help you top up those skills too.
Our first topic is homeostasis and response, and we're starting with number 1, homeostasis.
Okay, let's start with a definition. Homeostasis is regulation of the internal environments or conditions. That just means keeping things constant. This is both inside cells and inside whole organisms, like this little fishy. So it's important to keep these conditions constant so that enzymes work effectively. These are inside cells and they're easily affected by changes in pH and temperature. So we need to keep the conditions optimal, which just means at their best. So there are 3 conditions that you need to know in humans. The first is the blood glucose concentration. The second are the water levels. And the third is body temperature, which is around 37 degrees Celsius in humans. These all need to be kept at a constant. So a stimulus, this is a trigger for the homeostatic processes in the body, and it's just a change in the environment. An example of this could be on a hot day, the temperature rises and your body feels hot. You need to detect that change, and receptor cells are what do that detecting. Once you've got that information, it needs to be passed on to a coordination center to be received and processed. 2 examples of this are the brain and the pancreas. These decide how to respond. Effectors then actually carry out the response, and they restore the conditions back to the optimal level. The 2 types you need to know are muscles and glands, such as the pancreas.
2. The human nervous system.
In the homeostasis video, we learned that a stimulus is a change in the environment and that these changes are detected by receptor cells. There are 5 main sense organs in the body. Pause and see if you can name them and what they detect. Alright, so the eye detects light, the nose detects chemicals, this is by smell. The tongue also detects chemicals, but this time it's more by taste. The ears detect both sound and balance, basically being level. And the skin detects pressure and temperature. Once a stimulus is detected by receptor cells, nerves will carry this information to the central nervous system, or the CNS. This is made up of the brain and the spinal cord. Before we look at the journey through the nervous system, you need to know what neurons are. These are individual nerve cells. Here's a picture of one. It's not very good, but you'll see some on the next page that are a little bit better. If you have loads of them bundled together, say hundreds or thousands, then this is actually called a nerve. So that's the difference. A neuron is just one nerve cell. Okay, now let's take a look at the path of the nervous system. So, a stimulus occurs. The first thing that happens is the receptor cell detects that stimulus, that change in the environment, and this causes an impulse to then be passed onto a sensory neuron. This passes the impulse to a relay neuron and then to a motor neuron. So we've got 3 neurons. Finally, the motor neuron will then trigger the effector to carry out a response. Let's look at an example. So here our stimulus is some cold ice cream dripping onto a finger. The skin cells in the finger are going to detect that, and they're going to trigger an electrical impulse to be passed on the sensory neuron. This then passes onto the relay neuron, which is in the central nervous system, the spine and the brain. The impulse passes through the motor neuron and then to the effector. In this example, the muscle in the finger would move away from the dripping ice cream. In reality, lots of other responses could also happen, like salivary glands releasing saliva in preparation for licking the ice cream. Now, just to point out that this is a one-way system, and because it's happening through electrical impulses, it is very, very fast. Okay, speaking of impulses, we're going to point out a little bugbearer. We don't call them messages. Examiners hate that. We must call them impulses. Another tip is how to remember the relay neuron is like a relay race. So if you think about it, in a relay race, you pass on a baton from one person to another. So it's passing between different people. A relay neuron also just passes an impulse between different neurons.
3. Reflex actions.
Although the nervous system is very fast, there is something slowing it down: synapses. If you look very carefully, you'll notice there is a tiny gap between these two neurons. This is called a synapse. Here is a zoomed-in version of a synapse. As the electrical impulse reaches the end of one neuron, it causes chemical messengers, also known as neurotransmitters, to be released and diffuse across the gap. They attach to receptors on the next neuron. This causes a new electrical impulse to be triggered, and it will then travel across that neuron as well. The diffusion of the chemical messengers is slower than the electrical impulse. It's important to say that the chemical messengers diffuse, not travel, across the synapse. And just a reminder, we don't call the impulse a message, we have to call it an electrical impulse. In certain situations, we need a very quick response. Here, the person is holding their hand close to the fire. Because they're too close, their hand will automatically pull away, without them even realizing that they're doing it. This is called a reflex action. It happens automatically and rapidly. It does not involve conscious parts of the brain. But the main steps are the same as other nervous system responses. Pause and see if you can name each part in this sequence. Okay, so we start with receptor cells, then the sensory neuron, the relay neuron, the motor neuron, the effectors, and finally, the response. If you need to recap this, go back to my nervous system video. Okay, so let's look at this example. The receptor cells here are going to trigger a response to the high temperature of the fire. This travels along the green sensory neuron, up to the spinal cord, and then to the relay neurons. But note that the relay neurons don't go all the way up to the brain. They're going to come straight back and connect to a motor neuron. And the effect is then that the muscles in the hand will contract, pulling the hand away from the danger. Reflex actions are important because they help protect an organism from harm. Many basic body functions are also reflex actions, like breathing. If the brain were consciously involved in these actions, you wouldn't be able to think about much else.
4. The endocrine system.
We learned about the nervous system in a previous video, and now we're learning about the second system, the endocrine system. This is composed of glands, like the ones you see here. Glands secrete hormones into the bloodstream. They travel through the blood to their target organ, where an effect is actually produced. Traveling through the blood is a lot slower than the electrical impulses of the nervous system, but the effects are longer lasting. You need to know the names and positions of 6 glands in the human body. Pause and see if you can name any of these. Alright, we've got the pituitary gland up here beside the brain. Then we've got the thyroid gland. This is the pancreas. These are the adrenal glands. And in males, we've got the testes. And in females, we've got the ovaries. Let's come back to the pituitary gland. This is called the master gland. This is because it releases several hormones that can travel to other glands and cause them to release hormones. A tip to remember that it is the master gland is that it is found beside the brain, which is also like the master of the nervous system. The word pituitary is a rather long, fancy sounding word, which I think sounds a bit like an olden day master's name. The next bit of info we're looking at is higher tier only, so if you're studying foundation, skip ahead to the questions. Okay, so we're going to look at 2 glands and the functions of the hormones that they produce. So we'll look at the thyroid and the adrenal glands. First up, the thyroid gland makes thyroxine. This controls the basal metabolic rate in your body, which is basically how quickly substances are built up and broken down. It controls growth and development in children, but in adults, the levels of thyroxine stay pretty constant. Next is adrenaline, produced by the adrenal glands. What do you think it does to these body parts? Well, this hormone increases the heart rate. It also increases the amount of oxygen and glucose supplied to the brain and the muscles. And finally, it increases your breathing rate. These changes prepare you for the fight or flight response. Say you see a wild boar in the forest and it charges at you, you might try to fight it or you might run away. So, adrenaline is produced in stressful situations. When the situation is over, the adrenaline levels naturally lower. Now we're going to look at something called negative feedback. It controls different hormones and conditions to keep them at a constant level. And we're going to look at thyroxine. So the pituitary gland makes something called TSH, or thyroid stimulating hormone. It makes the thyroid gland make more thyroxine. Let's say that the thyroxine levels increase. The response will be that the pituitary gland makes less TSH. And less TSH means the thyroid gland makes less thyroxine. This will bring the thyroxine levels back to the set point. On the other hand, if the thyroxine levels decrease, then the pituitary gland will need to make more TSH. More TSH means that the thyroid gland produces more thyroxine, again bringing it back to the set point.
5. Control of blood glucose.
Glucose is essential for our cells. The mitochondria in our cells use it to respire. This releases energy for essential functions like keeping warm, moving, and building larger molecules. If we don't have enough glucose, our cells won't be able to respire. If we have too much glucose, it can damage organs like our eyes and our kidneys. We need to keep blood glucose levels within a small range. Let's look at what happens when we eat a meal. This bowl of spaghetti contains carbohydrates. These are broken down by the enzyme amylase in our digestive system to make lots of glucose. This glucose will then travel in our bloodstream. The organ that detects any change in glucose is this one, the pancreas. If the blood glucose levels are too high, it will release a hormone called insulin, which also then travels in the bloodstream. It allows glucose to move out of the blood and into our cells, where it is used for respiration. If there is excess glucose that is not used, it is stored as glycogen in both the liver and the muscles. This glycogen is insoluble, which means it doesn't dissolve, so it can be stored. This restores the blood glucose levels back to normal. And when the blood glucose levels are low, the glycogen can be turned back into glucose. If you're studying higher tier, watch on to find more about this. If you are studying foundation tier, just skip ahead to the questions. Okay, let's look at what happens when blood glucose levels drop. This could happen due to exercise, because your cells are respiring faster, using up more glucose. So let's imagine we have less glucose in the blood. It is still going to be the pancreas that detects the fall in the blood glucose levels, but this time it produces a different hormone. We'll use blue to illustrate it. And this is called glucagon. Again, glucagon travels in the blood, but it travels to the liver. That's its target organ. And here, it turns the stored glycogen back into glucose. This is then released back into the blood, increasing the blood glucose levels back to normal. Alright, it's important that you learn these spellings: glucose, glycogen, and glucagon. It's really important to make sure you've got them clear in your head, because if you spell them wrong or mix them up, you're going to lose the marks. One way to remember what glucagon does is realizing that it sounds like glucose gone. So it is a hormone produced when glucose is gone from the blood, or at least it's in lower levels.
6. Treating diabetes.
There are two types of diabetes. You need to be able to compare and contrast them. We'll look at what the disorder is, the causes, and the treatments. Okay, so in type 1 diabetes, the pancreas doesn't actually produce any insulin. This means that the blood glucose levels can rise to fatally high levels. For the cause of it, we know that it has a genetic element. We also know that it tends to start in young children and teenagers. The way we treat it is with insulin injections. No, they're not as big as it looks like in this picture. Okay, these are given before meals, so the insulin is in the blood, ready to deal with the increasing blood glucose levels. At the moment, there is no cure for type 1 diabetes, so people with this disorder will have to take insulin injections for the rest of their lives. But there are lots of new advances in how this insulin is administered. Okay, let's look at type 2 diabetes. This is when the pancreas does make insulin, but the body cells just don't respond to it. The main risk factor for type 2 diabetes is obesity, but other factors include old age and a lack of exercise. Treating it then seems a little obvious. People with this disorder need to do more exercise. They also need to eat a carbohydrate-controlled diet. Remember, carbohydrates are broken down into glucose, so you need to regulate those to make sure the glucose levels don't rise too high. These are the two main ways to regulate your blood glucose levels and control type 2 diabetes. However, if this isn't working, doctors may resort to drugs. These can influence the cells to be easier able to take up the insulin or for the pancreas to make more insulin. But again, if that really doesn't work, they will resort to insulin injections as well.
7. Reproductive hormones.
During puberty, reproductive hormones cause secondary sex characteristics to develop. In males, the testes make the main reproductive hormone, which is testosterone. This stimulates sperm production. Other changes include a growth spurt and the development of underarm, facial, and pubic hair. The voice will also break, which just means that it gets a little bit deeper. In females, the ovaries stimulate the production of the hormone estrogen. Other changes involve a growth spurt, the development of underarm and pubic hair, and the breasts start to grow. They'll also begin their menstrual cycle. If you're studying higher tier, you're going to learn a lot more about the hormones in the menstrual cycle, but everybody needs to know what they are and what they do. So we're going to start with that, and if you're doing higher tier, make sure to watch my menstrual cycle video. Let's start with seeing if you can label the parts of the female reproductive system. First up, we've got the ovary, which is joined to the oviduct, or the fallopian tube. This is the uterus, this is the cervix, and this is the vagina. Okay, so there are 4 hormones that you need to know about that control the menstrual cycle. The first is FSH, which stands for follicle stimulating hormone. This causes one egg in the ovary to mature every 28 days, which is called ovulation. Here's our mature egg. Luteinizing hormone, or LH, stimulates the release of the mature egg. Up here, we can see that it will travel out of the ovary and into the oviduct. Estrogen and progesterone carry out a similar function. They help to maintain the uterus lining, which just means it stays thick, ready for a fertilized egg to come along and implant into.
8. The menstrual cycle.
This is higher tier content, so if you're studying foundation, skip to the contraception video. First, you'll need to know the stages of the menstrual cycle, then we'll look at the hormones behind it. It's a 28-day cycle, and we'll start with the egg maturing in the ovaries, just like you can see here. The uterus lining also thickens in preparation for a fertilized egg. On day 14, one egg is released from an ovary. This is called ovulation, and it passes into the oviduct. The uterus lining will remain thick for a few more days after this, but if the egg doesn't get fertilized and implant, then the lining will break down. This is known as having a period. When this happens, blood will come out through the vagina, and a girl will need to use some sanitary products to capture it. She can use pads, tampons, or menstrual cups. Now let's look at the 4 hormones that control the menstrual cycle. FSH, or follicle stimulating hormone, comes from the pituitary gland and travels down to the ovaries. It causes eggs to mature. It also stimulates estrogen production. Estrogen is secreted by the ovaries. It causes the lining of the uterus to develop, ready for a fertilized egg. It inhibits or stops the production of FSH, so eggs do not continue to mature. It also stimulates the release of LH. LH, or luteinizing hormone, causes ovulation. This causes the mature egg to be released into the oviduct. Finally, progesterone is produced by the empty follicle in the ovary after ovulation. It inhibits LH, as you don't want more than one egg to be released. It also inhibits FSH. Finally, it maintains the lining of the uterus for around 10 days, keeping it thick and ready for a fertilized egg.
9. Contraception.
Methods of contraception can be split into hormonal and non-hormonal. We'll start with the hormonal methods, and as you need to be able to evaluate the different types of contraception, we'll look at advantages and disadvantages of each. First up, we've got the oral contraceptive pill. This is taken every day and contains hormones that will stop the egg from maturing. Do you remember which hormone normally makes the eggs mature? Yeah, it's FSH. So the pill inhibits FSH, which just means it stops the production of FSH. The big advantage of the pill is that it's easy to use, and if it's used properly, it's highly effective. The disadvantage is it can be really easy to forget to take it, or if you vomit, you can lose your pill for the day. And it has some side effects like increased blood pressure and other things. Next methods are the patch, the injection, and the implant. All of these use the hormone progesterone. This inhibits eggs in the ovary from maturing or from getting released, and they last progressively longer. So this is a big advantage of these methods. However, they all need replacing, and the injection and the implant require a nurse or a doctor to receive. Now we're going to look at the non-hormonal methods of contraception. First up, we've got spermicides. This is a chemical method that can kill or stop sperm from actually working. The benefit of these are that they're readily available. You can buy them from any pharmacy. But the disadvantage is they're not very effective and shouldn't be used on their own. Next up are barrier methods, such as condoms for males and diaphragms for females. The good thing about these is that there are no side effects and they can prevent against some STDs. The disadvantage is they need to be fitted correctly, and if they get damaged, they can easily let sperm through, increasing your risk of pregnancy. Here we've got an IUD, or an intrauterine device. This is inserted into the uterus, just like you can see over here. It works by preventing early embryos from implanting into the uterus lining. Some of them will also contain progesterone. The big advantage is that it's very effective, and once inserted, it's obviously very long-lasting. However, in the early days, it can be uncomfortable and cause some period pains. Next up, we've got surgical methods. This is for males in the sperm duct, and in females, the ovary. These can be either cut or tied. The big advantage is that it's permanent. This is for people who know they don't want to conceive. The disadvantage is that for women, this procedure requires a general anesthetic, which carries a risk with it. The final method is abstinence. This is making sure that you don't have sexual intercourse when the egg is in the oviduct. The advantage to this is that it's got no side effects whatsoever, but the disadvantage is a big one. It's really risky and you have a high risk of getting pregnant with this method.
10. Infertility treatments.
The NHS states that as many as 1 in 7 couples have difficulty conceiving or getting pregnant. This is called infertility. It can be caused by blocked or damaged oviducts, or a lack of ovulation. In males, a low sperm count or sperm defects, like having a shortened tail, can also cause infertility. Being obese or anorexic and simply getting older all increase your risk of infertility. If a woman doesn't naturally ovulate because her body isn't producing enough FSH or LH, this can be treated with fertility drugs. These contain the right doses of these hormones. Then she can try to conceive naturally. IVF, or in vitro fertilization, is another treatment that can be used for women who have faulty oviducts or when there's no obvious cause for infertility. The first stage is exactly the same. She is given fertility drugs. This causes her eggs to mature, and these are then removed from the ovaries, like this. Then the eggs are mixed with sperm in the lab, so that fertilization happens. If the infertility was caused by the man not producing enough sperm, individual sperm can be ejected into the egg. Microscopy can then be used to monitor that fertilization has actually happened, and to check that the fertilized eggs develop into the embryos, which are just tiny little balls of cells. 1 or 2 embryos are placed into the mother's uterus, bypassing the faulty oviducts. Hopefully, at least one baby will grow and be born. Although fertility treatment gives a woman the chance to have a baby on her own, it is very emotionally and physically stressful. Placing 2 embryos in the uterus can also lead to multiple births, which is a higher risk to the babies and the mother. Finally, the success rates are not high, and these decrease with age.
Now we're on to topic 6, inheritance, variation and evolution. And we're starting with 1, sexual and asexual reproduction.
There are 2 types of reproduction, sexual and asexual. Let's start with sexual. This involves fertilization, which is the fusion of the male and female gametes. In animals, the male gamete is the sperm cell, and the female gamete is the egg cell. But in flowering plants, the male gamete is actually pollen, but the female gamete is still an egg cell. When the gametes fuse together, there's mixing of genetic information from the male and the females. This leads to variation or variety in the offspring. Can you spot some differences in these 3 little hedgehogs? Now let's look at asexual reproduction. Asexual reproduction involves one parent only. The parent cells reproduce by mitosis to make the offspring, so there is no fusion of gametes, or no fertilization. Since there's no fertilization, there's no mixing of genetic information. So the new offspring, like this little runner coming off the strawberry plant, is genetically identical to the other runner. Because of this, we call them clones. They are all the same.
2. Meiosis.
In the previous video, we learned that sexual reproduction involves the fusion of gametes, and that gametes are made by the process of meiosis. We're going to learn what meiosis is in this video. Okay, first up you need to know that humans have got 46 chromosomes in their body cells. These are arranged in 23 pairs, or we say they have 2 sets of chromosomes. In gametes, like the egg cell and the sperm cell, they have 23 chromosomes. They're not arranged in pairs. We say they have 1 set of chromosomes. Meiosis is how gametes are made. Let's look at the definition. Meiosis is a type of cell division that happens in the reproductive organs to have the number of chromosomes, making gametes. So it happens in the testes and the ovaries. Let's take a look at the actual process of meiosis. Here we've got a body cell, so we'll give it some chromosomes. The first stage is the same as mitosis. The genetic information doubles, or copies, so we get twice the number of chromosomes. The next stage is the same as well. We have a division. So this cell splits in 2, and the genetic information is split between them. Okay, here's where meiosis differs from mitosis. It has a second division. So we're going to end up with 4 cells. Each one is a gamete. And each one has got half as many chromosomes as the parent cell, or the body cell. Let's look at this in terms of humans. In the body cell, you've got 46 chromosomes. So when the genetic information is copied, you end up with 92. Then, in the first division, where the cell splits in 2, you're going to go back to having 46 chromosomes in each cell. And in the second division, when each cell divides in two again, you'll get 23 chromosomes in each gamete. Okay, let's look at what happens when the gametes fertilize. The egg and the sperm each have 23 chromosomes, so when they fuse together, you're going to get the full 46 chromosomes in the fertilized egg cell. The fertilized egg cell will then divide, this time using mitosis. It does this many times to form the early embryo, and each cell of the early embryo will contain the 46 chromosomes. As the embryo develops, the cells of the embryo differentiate. This just means that they become specialized for a particular function, like detecting light in the eyes. Eventually, all the cells are specialized and you have a baby. The last thing to mention is that meiosis makes every gamete unique, so every offspring is different.
3. DNA and the genome.
Inside our cells, the nucleus contains chromosomes. In the meiosis video, we learned that our body cells have 46 chromosomes. Every chromosome is made up of DNA, and DNA is made up of lots of small units, so we can call it a polymer. It is made from two strands that form a double helix. This is a definition you need to learn. Now let's look at genes. A gene is a small section of DNA that codes for a specific sequence of amino acids, and these make a specific protein, for example, the pigment that colors your eyes. You'll learn a lot more about how genes work in the next video. As I mentioned at the start, scientists have sequenced the human genome. But what is a genome? Well, it is simply the entire genetic material of an organism. This includes all the DNA inside the nucleus and the DNA found inside the mitochondria. Understanding the human genome is an important area of research. Scientists are using it in 3 ways. Number 1, they're using it to improve their understanding of inherited disorders and how to treat them better. This includes looking at cystic fibrosis and sickle cell anemia. Number 2, they're using it to search for genes linked to disease. This includes diseases like heart disease and type 2 diabetes. To do this, they're looking for patterns in the genomes and trying to spot any similarities. And finally, number 3, they're using genomes to trace past human migration patterns to look at how we moved across the world.
4. Inheritance keywords.
In the DNA video, we learned that genes are sections of DNA that code for proteins. Some characteristics are controlled by a single gene, such as fur color in mice and red-green color blindness in humans. But most characteristics are as a result of multiple genes interacting, rather than a single gene. For example, height in humans. Some genes have different forms. We call these alleles. For example, the gene for fur color in mice has got both a brown fur allele and a black fur allele, as well as some others. Alleles operate at a molecular level to develop characteristics. We call these physical characteristics phenotypes. So it's just the appearance of the organism. Here's a hint. Use the pH to help you remember that phenotype is physical characteristics. Genotype is then just the alleles that an organism has inside it. Remember, an allele is just a different form of a gene, so the geno part of the word just means alleles. Let's take a look at how alleles work. We'll look at the example of fur color in mice. Looking at these alleles and the word homozygous, can you work out what homozygous actually means? Pause, have a think, and press play when you're ready. Homozygous means having 2 of the same alleles. Here's a hint. You might have heard the word homosexual when a person is attracted to the same gender as themselves. So homo simply means same. What then do you think heterozygous might mean? Heterozygous means having two different alleles. And by the same token, being heterosexual means a person is attracted to a different gender than themselves. So hetero just means different. And you can see these are different. But what fur color or phenotype will the mouse have? We need to know more about the alleles. There are two types of alleles. Dominant alleles are always expressed or shown, whereas recessive alleles are only expressed or shown if there are two copies of that allele, i.e., both of the alleles are the same. We represent dominant alleles with capital letters, whereas recessive alleles are shown by lowercase letters. You'll get told which allele is dominant and which is recessive, so you don't need to memorize that. In this case, black fur is the dominant allele, so it gets the big B, and brown fur is recessive, so it gets the small b. So let's look at the genotype of this mouse. It's got the big B for the black fur, and then it has a little b for the brown fur. Note that the capital always goes first. Since the dominant allele is for black fur, the mouse will have a phenotype of black fur. If we look over here, we can practice our genotypes. This mouse has two dominant alleles, so we write it as big B, and the phenotype is black fur. But this mouse has two recessive alleles, so we write it as little b, and the phenotype is brown fur. We can call the genotypes homozygous dominant if there are two dominant alleles, and homozygous recessive if there are two recessive alleles.
5. Inheritance Punnett squares.
In the last video, we learnt a lot of the keywords needed to understand inheritance. Now we know how alleles work, we can use them to predict the genotypes and phenotypes of children or offspring. Let's look at eye color in these parents. The mum's phenotype or physical characteristic is blue eyes, and the dad's phenotype is brown eyes. For the genotype, we need to know which allele is dominant and which is recessive. Remember, you'll be told this or will be able to work it out from the information given. In this situation, the blue eye allele is recessive. So we're going to label it with a lowercase letter, a little b. And so the mum's genotype is little b. The dad has a dominant allele, which gets a capital B. So his genotype is big B, little b. But if they have a child, how do we know what genotype and phenotype it can have? Well, to do this, we're going to use Punnett squares. This is just a genetic diagram, and to start off, it looks a little bit like a noughts and crosses grid. We'll start by splitting up the mum's alleles, because a child only gets half of its genes from its mother. So we split up the little b and the next little b. We'll do the same for the dad, for the same reason that the child gets half its DNA from his dad as well. So we split up the big B, little b. Now we work out the combinations by reading down and across. And we get big B, little b. In the next box, we get another big B, little b. Why not pause and see if you can work out the remaining two boxes? In this one, we get little b, and it's the same in the last box. Great, so now we have all the genotypes. We'll work out the probability of each. You could be asked to give this as a fraction or a percentage, so we'll do both. Okay, so you can see we've got two big B, little b's, and two little b's. That means we've got 50% of each, or a half. So we write the letters out, big B, and we do the same for the little b. Now we'll write the phenotypes for each genotype. You need to write them below in the same order, or beside the genotype so it's very obvious. Exam questions will normally structure this for you. So the big B, little b will give you brown eyes, as the brown eye allele is dominant. The little b will give you blue eyes, because you have two recessive alleles. Lastly, you can be asked to give the expected ratio. This is one of the math skills you're required to know for your exam. Since it's both 50%, as a ratio, this is 1 to 1. Again, you write it in the same order as you've given the phenotypes. If you're studying higher tier, you need to be able to make the Punnett squares yourself. If you're studying foundation, you need to be able to complete one, and the questions will be more structured. I recommend you watch this either way, as it's good practice. Okay, let's read the question. Two smooth pea plants are crossed. They are both heterozygous. Work out the proportions and ratios of the genotypes and phenotypes for this cross. Cross just means they're reproducing together. Pause and grab some paper and try and work it out yourself. Then press play when you're ready to go through it. Okay, so let's start with the parent genotypes. They are both heterozygous, which means they have different alleles. The two alleles we've got are smooth is dominant and wrinkled is recessive. We'll use the letter S. The dominant one gets a capital and the wrinkled one gets a lowercase. It doesn't matter what letter you use, because we haven't been given it. So both parents' genotypes, because they're heterozygous, will be big S, little s. Now we're going to do our cross. So we start by splitting up each parent's genotype into big S, little s. Then we work out the different combinations. If you didn't get to this point, pause anyway and try and fill out the combinations. Okay, so this box will be big S, this is big S, little s, another big S, little s, and two little s's. Notice how I'm really emphasizing the size of the letters. That's important. Okay, now let's write out what offspring genotypes we have. We can see we've got one big S, which is 25%. We've got two big S, little s's, which gives us 50%. And we've got one little s, which is 25%. The question has asked us for the ratios of the genotypes. So if we work it out, we've got 1 to 2 to 1. If you wrote your genotypes in a different order, you may have your ratio in a different order. Just check carefully. Next, we'll work out the offspring phenotypes. Every offspring that has a dominant allele, or a big S, is going to be smooth. But every offspring that has two little s's is going to be wrinkled. We can see that we've got three offspring that have got the big dominant S. So 75% are going to be smooth. And that leaves 25% will be wrinkled. Finally, we have to give this as a ratio. And we compare 75 to 25, that's 3 to 1. The last thing we're going to look at is sex inheritance. This person's phenotype is female, and this person's is male. Sex is controlled by an entire chromosome, as opposed to one or multiple genes. In females, the sex chromosomes are XX, and in males, they are XY. We can construct a Punnett square to show the offspring probabilities. Doing this very quickly, we can see that we end up with two XX offspring and two XY offspring. So the probability is 50% female and 50% male. You have to memorize the sex chromosomes for females and males. One tip I use to help me remember this is that my female friends tend to send me a little kiss kiss, like an XX, at the end of messages, more than my male friends do.
6. Cystic fibrosis and polydactyly.
People who have the genetic disorder polydactyly will have extra fingers or toes. Part of the word poly actually means many, like a polygon is a many-sided shape. It is caused by a dominant allele. This means people only need to have one allele to get the disorder. Cystic fibrosis is a disorder of the cell membranes. The cell membranes struggle to move substances around, which leads to thick mucus building up in places like the lungs. It is caused by a recessive allele, which means you need two recessive alleles to have the disorder. If a person just has one of the recessive alleles, they don't have the disorder, but we call them carriers. You may be asked to use Punnett squares to work out the probabilities of offspring inheriting these disorders. We'll start with a parent that has got polydactyly because they've got the dominant allele for it. We'll then have them crossing with another parent that doesn't have polydactyly because they don't have the dominant allele. Let's do our combinations by reading down and across, and then we'll work out what genotypes we've got. So we can see that we've got 50% big D, little d, and we've got another 50% of little d, little d. The phenotypes, or the physical characteristics, are then 50% polydactyly, because they've got the capital dominant allele, and 50% normal. And as a ratio, this is 1:1. Now let's look at a cystic fibrosis example. Let's start with a person that hasn't got cystic fibrosis, and they have no cystic fibrosis alleles. Then we'll have another parent who does have cystic fibrosis. When we do our combinations, we can see that for our genotypes, all of the offspring would have big C little c, 100% probability. As big C little c has a normal dominant allele, they are 100% normal or non-cystic fibrosis. Couples with a history of a genetic disorder in their family may be offered genetic screening. This is when cells from the embryos are taken and screened or tested for specific disorders. If the tests show that the embryo has a genetic disorder, the parents will make a decision whether or not to keep the baby or whether to terminate the pregnancy. There are, of course, many complex issues with this. You need to be able to make judgments using information given, so don't worry too much about memorizing these. First of all, with economic issues, is that screening is very expensive and currently only offered to those with a family history of disorders. Some people think every couple should be offered it, while others think the money could be better spent somewhere else. However, supporting a family with a child affected by a genetic disorder is also very expensive. Now we'll look at the ethical issues. Collecting cells from an embryo can increase the risk of a miscarriage, whether or not the baby has the disorder. And occasionally, the test can give a false positive, saying the baby has a disorder when it hasn't. It can also be a false negative, saying the baby doesn't have the disorder, but when it is born, it does. And of course, once a couple has the screening done, they face their own ethical decisions on whether or not to have an abortion. Finally, social issues affect us all. People are concerned that genetic screening could create a demand for designer babies, which is when they're chosen for their certain characteristics.
7. Family trees.
Family trees are used to show inheritance. This is often used whenever there is a family history of a genetic disorder, like cystic fibrosis. You will be given a key to use to figure out some answers. Here's an example. Males and females will be given different shapes. Shading is then used to indicate if the person has got the disorder. Okay, so here we've got a man and a woman. When they're joined by a horizontal line, this means that they are parents. A vertical line with branching other symbols means that they've got children. So we can see these parents have 3 children. If they're adult children, they may have a partner of their own and form parents together. The best way to understand family trees is to give some questions a go. So pause and try them and then press play when you're ready to go through them. So always start by looking at the key. Okay, number 1. What is the genotype of an affected male? Well, this is a cystic fibrosis family tree, and as we learned in the last video, cystic fibrosis is caused by recessive alleles. So we need 2 recessive alleles to have the disorder. Lizzie and Amir have a healthy daughter, Eva. What symbol would she have on the family tree? The key here is she is a healthy daughter, so we're looking for an unaffected female. So we're going to give her a blank circle, and if you can, use black. Okay, what is Eva's genotype? Genotype just means what alleles she has. So we already know that she's unaffected, which means she has a normal dominant allele, which she got from her dad. But her other allele will have come from her mother, who has got cystic fibrosis. and therefore she can only pass on a little c to Eva. So Eva's genotype is big C, little c. 4. What is Sarah's genotype, and explain how you know this? So let's find Sarah. Here she is, and she's also an unaffected female. So because she's unaffected, she must also have a big C or dominant allele. But how do we know what the other allele is? Well, we need to look at her children to see what she and Tom have passed on. One of her children has cystic fibrosis. One allele came from Tom, but the other must have come from Sarah. This means that Sarah must have a recessive allele herself, as she passed it on to Lizzie. Try to use names in your answers if they've helped you work out the answer. So she's big C, little c. And finally, Kira and Alex are both carriers of cystic fibrosis. How does the family tree provide evidence for this? So just a reminder, carrier means they've got one recessive allele and one normal allele. So they're both big C, little c. But how do we have evidence for this? Well, we can see that they've got children that are affected or have cystic fibrosis. But if we look at them, we can see that they haven't got the disorder themselves, which means they must be just carriers. All right, how did you do in the questions?
8. Variation. Variation is defined as the differences in characteristics of individuals in a population. This is a definition you should learn. So for example, poodles have got curly fur, while Maltese have long straight fur. St. Bernards have floppy ears, while German Shepherds have ears that go really pointy and upright. These are all examples of variation within dog populations. Can you think of any examples of human variation? Now we're going to look at what causes variation. First up are genetic factors. This just means that the genes you inherit from your parents can determine some of your characteristics, like natural eye color. There is a lot of genetic variation within a population. This is caused by mutations. Mutations are changes to your DNA, and they're happening all the time, but it's very rare that they actually lead to any physical change. But when they do, you get a new variant. Variation can also be caused by your environment. This means any external influences that are changing your characteristics. For example, just dyeing your hair. This changes the color of it. Another example are hydrangea flowers. If the plant is in acidic soil, the flowers will be blue, but if it's put in alkaline soil, the flowers change to pink. And finally, there can be combined causes of variation. And this is where variation is caused by both your genetics and the environment interacting together. For example, skin color. You naturally inherit this from your parents, but if you were to spend all day sunbathing, your skin would darken. Most variation is caused by both your genetics and your environment. Other examples include height and weight. 9. Evolution by natural selection. Evolution is a change in the inherited characteristics of a population over time. For example, these plants have purple flowers at the start, but over many years they may evolve to have blue flowers instead. Evolution happens by the process of natural selection. This theory states that all species of living things have evolved from simple life forms that first developed over 3 billion years ago. That's 9 zeros. It's a long time, but remember, this is making every species. Okay, so you need to know the stages of natural selection. So let's break it down. It starts with mutations. These are changes in the DNA that cause variation in a population. Most of the time, mutations don't do anything and just go unnoticed, but very rarely they can cause a new phenotype or a new characteristic. For example, a mutation could cause this caterpillar to be brown rather than green like the rest of the population. This might be bad news because he stands out to predators, but maybe there's a change in the environment. For example, if there was drought, the leaves might be brown as well. Suddenly, being brown's looking pretty good, as he's really camouflaged. So if this new characteristic is beneficial, the individuals are more likely to survive and breed. When this happens, they're going to pass on that beneficial allele to their offspring. This will happen again and again until all of the population possesses the beneficial allele. We say that the population has evolved. If two populations from the same species become so different in their phenotype that they can no longer breed successfully to produce fertile offspring, like the green and the brown population of caterpillars, we say we've got a new species. 10. Selective breeding. Selective breeding is the process by which humans breed animals and plants for particular genetic characteristics. There are 4 examples in the specification to learn. First, we've got disease resistance in crops, animals which produce more meat or milk, domestic dogs with gentle natures, and large or unusual flowers that will sell well. The first cases of selective breeding were thousands of years ago, when humans first bred food crops from wild plants and they domesticated animals. To do selective breeding, there are a few stages. First of all, you must select the parents that have the desired characteristics. For example, maybe floppy-eared bunnies are more popular pets, so you would select 2 parents that have floppy ears. The second step is to then breed them together. They will produce lots of offspring, some of which will have the floppy ears. This is because the allele for the characteristic is not always passed on to every offspring, just some. The third step is then to select the offspring that do have the desired characteristic, i.e. the floppy ears, and then you breed them together. You repeat this for many generations and eventually all of the offspring will have the desired characteristic. There are some problems with selective breeding. It can lead to inbreeding. This is when some breeds have an increased risk of disease or inheriting genetic defects. For example, pugs are known to have an increased risk of eye diseases and they often have breathing problems. 11. Genetic engineering. In the last video, we looked at selective breeding. This is a slow process that takes many generations of reproduction to change characteristics. Genetic engineering is a lot faster, but it is more complicated and requires more regulation. In this process, genes from the chromosomes of one organism are cut out. For example, the glow-in-the-dark gene. These are then transferred to another organism's genome, like a cat genome. This can then be used to make a new organism that will have the desired characteristic, in this case, a glow-in-the-dark kitten. Genetic engineering is more commonly done in plants. We call the genetically modified plants GM crops. They are modified in a number of ways, such as being more resistant to both disease and insect attacks, increasing yield so that more of the same plant is grown in the same space. Like here you can see more tomatoes have been produced inside the same flower pot. They can be modified for producing bigger and better fruits, like these massive apples. And they can be modified to become herbicide resistant, which means that the weeds are killed, but the crops are fine. Genetic engineering can also be done in bacteria. For example, putting the human gene for insulin into bacteria. This means the bacteria can then make insulin that's used to treat diabetes. If you study higher tier, you'll need to know the steps of how genetic engineering is done, so we'll look at those now. But if you're studying foundation, skip forward to the benefits and risks of genetic engineering. First of all, the desired gene is selected and cut out from an organism's genome using enzymes. You can think of enzymes as a pair of scissors. Then a vector will be used to transfer the gene. Vectors can be bacterial plasmids or viruses. In this case, it'll be a plasmid, which is just a loop of DNA. The plasmid will be removed from the bacterial cell, and then it will also need to be cut open, again using enzymes. The desired gene, which in this case is insulin, is then inserted into the vector. The vector then transfers the gene into the new organism. In this case, the new organism is another bacterial cell. When it reproduces, all of the offspring will have the desired characteristic. So all of these bacteria cells will be able to produce insulin. This is then harvested and used to treat diabetes. Please note that if a gene is transferred into an organism like an animal or a plant, it needs to happen at a very early stage of its development, i.e. when it's still a very early embryo. You've seen a lot of benefits of genetic engineering. For example, we saw lots of GM crops that had really good characteristics. One more possible benefit is being explored by modern medical research. They're hoping to cure genetic disorders, for example, replacing cystic fibrosis alleles with healthy alleles. This would be a huge benefit. However, there are some risks and objections to genetic engineering. Some people are concerned that the effects of eating GM crops on our human health hasn't been fully explored. There's also a risk of the modified genes spreading to other wild plants and flowers. Some GM crops were initially created to be infertile, so it would have a huge impact if these genes managed to spread. But so far, this hasn't happened. And some people worry there's a desire to manipulate the genes of future children in order to change how they look. Overall, scientists agree that genetic engineering has had huge benefits, but it's important to continue to regulate its practice. 12. Fossils and extinction. Fossils are the remains of organisms from millions of years ago that can be found in rocks. It's important that you do say millions of years ago when you define it. You need to know the different ways that fossils can be formed. The first is when the conditions for decay are absent, for example, warmth and oxygen. This insect is trapped in amber, so will have no oxygen. Secondly, hard parts of the organism, like the skeleton and teeth, are replaced with minerals as they decay. And thirdly, fossils can be preserved traces of organisms rather than the organism itself, such as footprints, burrows, and rootlet traces, which gives evidence of plants. We can learn from fossils how much or how little different organisms have changed as life developed on Earth, but the record is incomplete. Let's look at why it's incomplete. Well, first of all, most organisms that die don't actually become fossils. Then early fossils have actually been lost by geological activity. For example, when the crust moves and is pushed down into the mantle, the fossils get destroyed. Then we think that there are lots of fossils still to be found. Finally, early forms of life were soft-bodied, which means they left few or no traces behind, as soft parts decay and cannot be replaced by minerals. This is why we cannot be certain about how life began on Earth. Now we're going to look at extinction. Extinction occurs when there are no remaining individuals of a species still alive. There are loads of examples of this, but the most commonly known ones are the dinosaur species. You need to know how extinction can occur, so let's look at the 5 methods. First of all is the introduction of new predators. If a new species of predator appears, it can quickly cause the extinction of its prey species. This happened when humans introduced predators on the island of dodos. Secondly, introducing new diseases can quickly cause extinction if the species has no immunity to it. Next up, there can be new, more successful competitors. If the new species competes for the same resources, such as food or space, then the old one may die out. In this case, the new plant gets all of the sunlight and all of the water, and so the old plant will instead die. Now we'll look at some physical changes. There can be changes to the environment over geological time. This just means a really long period, like millions and billions of years. For example, an ice age could cause the extinction of species that live in really warm climates. And finally, single catastrophic events, like an asteroid collision or a volcanic eruption, could cause a mass extinction. This is one theory for how dinosaurs died. 13. Antibiotic resistance. Bacteria can evolve rapidly because they reproduce at such a fast rate. Some bacteria are pathogens, which means they cause disease, while others are harmless to humans. Mutations of pathogens can produce new strains of disease. Some of these strains are resistant to antibiotics, and so when antibiotics are used, they don't get killed. Instead, they survive and reproduce. The population of the resistant strain will increase, and the new strain will then spread. This is because people have no immunity to it. It's a brand new disease. There is no effective treatment for these resistant pathogens. MRSA is an example of a resistant bacteria that you need to know about. The MR means methicillin resistant, which is the type of antibiotic it's resistant to, and this superbug can commonly be found in hospitals. It's really important that we prevent the rise of antibiotic-resistant bacteria, or we may live in a world where we cannot treat infections. But you might ask, why not just make new antibiotics? Well, new antibiotics are very expensive and very slow to develop. It can take many years and they're unlikely to keep up with the emergence of new strains. So we need to prevent them. Here are 3 ways how. First of all, doctors should not prescribe antibiotics if they're not necessary, i.e. if your illness is caused by a virus, in which case antibiotics don't work, or if it's not a serious illness. Secondly, if you are prescribed antibiotics, it's your duty as a patient to make sure that you complete the full course so that all the bacteria are killed and none survive to mutate and form resistant strains. This is really important as some people think they feel better and stop taking them, but actually they've still got pathogens in their bodies. And thirdly, antibiotics should not be used in agriculture. Some farmers use antibiotics to prevent their animals getting sick in the first place, but this greatly increases the risk of resistant strains developing. 14. Evolutionary trees. Evolutionary trees are made using current classification data for living organisms and fossil data for extinct organisms. You need to know how to interpret them. The species name are put along the top of the diagram. I've used letters here to keep it simple. The time frame will be given, usually in millions of years, and you read across from it until you reach the branches. Where 2 branches meet, it shows the last common ancestor between 2 species. Where the branches split is where speciation occurred. Species A and B and all of the other species evolved through natural selection. Branches that split recently are closely related species, like B and C. Whereas branches that split a long time ago are distantly related species, like A and D. You can be asked to collect data from evolutionary trees. For example, when did species D evolve? Just find the species on the diagram, read down until the branch splits, and across, and you'll get your answer. And species D was about 7.5 million years ago. If there is a species whose line doesn't reach up to the top, or to the 0 million years mark, like species E, then this shows us that that species went extinct. In the case of species E, we can read across and work out that it went extinct 2.5 million years ago. 15. Classification. In the 18th century, Carl Linnaeus developed a classification system that is still being used today. It groups living things based on their structure and characteristics. He came up with these levels of hierarchy, with the highest and biggest at the top. It goes kingdom, phylum, class, order, family, genus, and species. This is something you need to know in the correct order, so let's make a mnemonic for it. You should try making your own with something that's really personal to you. You just take the first letter of each word and make it into a silly sentence. Here's an example. Keep ponds clean, or frogs get sick. No. In the exam, you just write your letters out again and then revert them back to the scientific words. The last 2 levels of classification make up the binomial classification of a species, which is just its 2-part name. Remember, bi means 2. So if you're asked for the species name or binomial name, you always give the genus and the species part. For example, humans are Homo sapiens, and wolves are Canis lupus. You'll be given the information needed to work this out. Notice that the words are written in italics and the genus name always starts with a capital letter. There is now a new level of classification developed by an American scientist called Carl Woese. This new level is called domains, and it is above kingdoms. So we could modify our mnemonic to say, definitely keep ponds clean or frogs get sick. He and his team came up with this new classification due to improvements in biology. First of all, microscopes have really improved a lot over the years, and we can now see a lot more detail of the internal structures of cells. Secondly, our understanding of biochemical processes that happen in living things has also really progressed since the 18th century. Domains can be split into 3 different groups, and you need to know the names of them. First of all, we've got the archaea. These are primitive forms of bacteria that live in extreme environments, e.g. hot vent bacteria that can survive really high temperatures. The next domain we've got are bacteria, also known as the true bacteria. The third domain is eukaryota. You'll have learned about eukaryotic cells in the first topic of biology, and hopefully you remember the 4 kingdoms: fungi, protists, animals and plants. And if you're wondering about the prokaryotes, which you also learned about in that topic, then archaea and bacteria both encompass these type of cells. Finally, I've got a little tip for you. To remember Woese's name, we can split it up. Maybe he was woeful, which means sad, when he first saw Linnaeus's classification system and realized something was wrong, and now he's saying, "See, look, I fixed it." Maybe it'll help. Now we're on the last topic, which is ecology. Number 1, communities. Ecosystems are complex. They are made up of communities of living things interacting with each other and the non-living parts of their environment. Some examples of ecosystems include grassland, desert, and tropical rainforest. To survive and reproduce, organisms require a supply of materials from their surroundings and from the other living things there. For example, cows need grass, and plants need sunlight, water, and nutrients, as well as other things. If one species is removed, it can affect the entire community. For example, if penguins were removed, then the number of seals would decrease, which in turn would affect the numbers of polar bears and killer whales. We'll look at food chains in more detail in a later video. Because species survival depends on other species, we say that they are interdependent. Inter just means between, so interdependent is dependence between species. If species are in balance with each other, and environmental factors are also in balance, then we call it a stable community. In this community, the population size remains fairly constant because of all of the balance. 2. Biotic and abiotic factors. Abiotic factors are the non-living factors which can affect a community. There are lots in this diagram. I want you to take a moment to pause the video and see how many you can spot. Ready? First up is light intensity. Plants require light for photosynthesis, so where it is dark, plant growth is limited, which of course will affect the rest of the community. Next is moisture levels. These are really important for all organisms, as all life needs water to survive. The desert is really dry, as it doesn't rain very often, so there are much fewer organisms living there. Some insects, like slugs for example, need really moist environments too. Wind intensity and direction can affect the ability of plants to grow and anchor their roots, as well as affecting smaller organisms. Did you spot this one? Carbon dioxide levels affect plants, as it's a reactant for photosynthesis. And occasionally, it can affect animals like mosquitoes that detect their prey by high carbon dioxide levels. Maybe you spotted this one too. Oxygen levels affect aquatic animals like fish that use it to respire. The pH of soil and its mineral content affects plants. Some plants don't survive in very acidic or alkaline environments, and of course all plants need minerals to survive. Temperature affects many organisms, as most live within a small range of temperatures. This is why climate change is having such a severe and drastic effect on species all around the world. Now we'll look at biotic factors. These are the living factors that affect communities. Why not do the same thing and pause the video and see how many biotic factors you can now spot? Ready? Here we've got a new predator. These can really disrupt a community, and they can even cause extinction. Availability of food is also really important, as without adequate food, populations can starve to death. New pathogens can also devastate populations if they have no immunity to it. And finally, one species outcompeting another can cause numbers to decrease to the point where they are no longer sufficient to breed. You can see this bird has got loads of worms, while this bird has none. Woo. Notice that 3 of these biotic factors are the same as 3 of the 5 reasons for species extinction. Can you remember the other 2? I recommend making a mnemonic to learn all of the biotic and abiotic factors that affect communities. This is a really good way to help you revise. 3. Competition. There are many organisms in any given community, and they all require resources, for example food or territory. These things are limited, so competition takes place when each individual attempts to secure the resources it needs. Competition can take place between different species, for example, different tree species in a forest competing for light. It can also take place within a species. This is often more intense competition, as the individuals require exactly the same resources. Let's look at animal competition. First of all, animals compete for food. Here we can see seagulls fighting over some chips. This is a really common form of competition. Secondly, animals compete for territory, which is just space. Lions are very territorial, and there is usually just one adult male in each pride. Pet cats are territorial too, and most cat fights are as a result of overlapping territories. Thirdly, animals compete for mates, or sexual partners. There are lots of adaptations in the animal kingdom to win mates, including the amazing male peacock's feathers. Males will often fight each other as well for access to females. Now let's look at plant competition. Plants compete for light, growing as quickly as possible vertically to reach the sunlight, or having large thin leaves to increase the surface area and maximize light absorption. Plants also compete for space. Often they're very crowded, so they spread their roots, stems, and branches. And when they disperse their seeds, they try and get the seeds as far away as possible from the parent plants. This means that the seeds will have enough space of their own to grow. In this example, you can see that the dandelion is using wind to spread its seeds, but water and animal dispersal are other methods. And finally, plants compete for both water and minerals from the soil. Their roots absorb both of these, and so the roots will spread as much as possible to absorb what the plant needs. 4. Adaptations. Adaptations are features that allow organisms to survive in the conditions in which they normally live. A simple example is that fish live underwater, so they need gills to take in oxygen from the water. Adaptations can be structural, behavioral, or functional. We're going to take a look at some examples of each, so that you understand what these words mean. But you don't need to memorize the examples, as the specification says you'll be given appropriate information to allow you to explain how organisms are adapted. Structural adaptations are physical characteristics. For example, polar bears have fur to insulate them in a cold environment. Another example is that cacti have spines instead of leaves, to reduce the surface area so less water is lost in a hot and dry desert environment. Behavioral adaptations are how the organism behaves. For example, a lizard basking in the hot sun on a rock to raise its body temperature. Another example is that birds migrate to warmer countries in the winter. Functional adaptations are about how something works or functions. For example, some fish and frogs living in very cold environments have got antifreeze in their cells so that they don't freeze to death. And some animals only give birth in a certain season to maximize their offspring's survival. There is one group of organisms that you need to know about in slightly more detail. These are called extremophiles. These organisms live in very extreme environmental conditions. For example, they may live in high temperatures, high pressure, or high salt concentrations. Bacteria that live in hot deep sea vents are called extremophiles, and these are an example you need to know. 5. Food chains. Food chains are simple ways of showing the feeding relationships within a community. They always start with a producer. A producer is an organism that can use light to carry out photosynthesis to make oxygen and glucose. Producers are green plants, algae like seaweed, and phytoplankton. Primary consumers eat producers. Here, the field mouse will eat the grass. Secondary consumers then eat the primary consumers. This weasel will hunt and eat the mice. Because it kills other organisms to feed from, we call it a predator. We then call the mouse the prey. Tertiary consumers eat secondary consumers. These are often top predators or apex predators that aren't hunted and eaten by anything else. For example, polar bears or killer whales. There can be levels above this, but this doesn't happen too often, as energy gets lost as you go up through each different level. You'll learn more about this when you study pyramids of biomass. Now let's look in more detail at predator-prey feeding cycles. If the prey, like this mouse, has access to lots of food, then its numbers will rise as more individuals survive and reproduce. We'll use green to represent prey. But if the predator now has a larger food source, it will be more successful too, so its population will increase as well. And we'll use red for the predator. Of course, now there are more predators to eat the prey, so the prey numbers will start to drop again, and they'll drop back to roughly where they started. But as the prey numbers drop, so will the predators, as there are too many to get enough food, so some won't survive to adulthood to reproduce. This cycle will continue, with fewer predators meaning the prey population can start to increase again, and so on and so on. The cycle will constantly repeat itself if it's in a stable community. 6. Sampling, transects and quadrats. Sampling is done by ecologists to find the abundance of organisms, that means how many there are, and to see the distribution of organisms, which means where they are found. We're going to look at two methods that are used. These are both part of a required practical, so it's important to make sure that you understand them. Firstly, you can use quadrats. These are square frames that are laid on the ground. They may or may not have a grid inside them. You can then count the number of plants or slow-moving organisms inside them. For obvious reasons, they can't be used for large or quickly moving organisms. How many flowers can you count inside this quadrat? Well, we can clearly see that there are 5 that are inside the quadrat, and 1 that's part in and part out. It actually doesn't matter which way you count it. It could be 5 or 6, as long as you use the same method for every quadrat sample that you do. It's also important to take as many samples as possible. This makes sure that your data is valid, which means that it shows what is actually happening. You also need to do quadrat sampling at random. You can do this by blindfolding yourself and spinning before throwing, closing your eyes, or a better way is to split your sample area up into a grid and then use a random number generator to pick your sample sites. If this quadrat were representative of a community, how many flowers would you expect to find in 1 meter squared? Well, first of all, let's take a look at the size of this quadrat. We've been given the length of it as 0.25 meters. We want to know the area as it's meters squared, and as it's a square, we're just going to multiply that number by itself. This will give us, on a calculator, which you are allowed to use, 0.0625 meters squared. Now we need to see how many times smaller this is than 1 meter squared. So we do 1 divided by 0.0625, and that comes out at 16. So this quadrat is 16 times smaller than a 1 meter squared quadrat. Let's say we took our count to be 5. We would now just do 5 times 16 to see how many you get in 1 metre squared. And it comes out at 80 flowers per metre squared. Now let's say you took 7 samples with your quadrat, and these are the results that you got. You could be asked to process the data to give any one of these. So let's take a look at what each one means and work it out. The mean is just the average. So to do this, all you do is add up all of the numbers and divide by how many values you have. So in this case, we had 7 samples, so we divide by 7, and this would give us 4.2857. Questions will specify if they want it to 2 decimal points or 2 significant places, etc. In this case, we'll do it to 2 decimal points, and the 5 makes the 8 round up to give us 4.29. The median is the middle value. So we need to write all of our numbers out from smallest to largest, and then we just check which number is in the middle, and is 5. The mode is the most common value. In this case, it's 6, as we have 3 of those. And finally, the range is the highest value minus, or take away, the lowest value, which in this case is 6 minus 1 gives you 5. The second method of sampling is a transect. We're going to look at line transects. This is just a measuring tape that is stretched between 2 points with quadrats placed along it at regular intervals, like every 5 meters. It allows you to see the effect of an abiotic factor on the organism distribution, and you can also measure this factor along the transect. For example, light intensity reducing from a grassland into a woodland. And you might predict that there'll be a big change in the distribution and variety of organisms found, as there'll be different amounts of photosynthesis able to take place in each area. 7. Water, decay and carbon cycles. The water cycle is all around you. You see this being cycled all the time in the form of rain. Water is evaporated from the Earth's surface to become water vapor. As it rises in the atmosphere, it cools down and will eventually condense. Then it falls as precipitation in the form of rain, snow, or hail. And then the cycle starts all over again. Next up is the decay cycle. Decomposers are really important. These are microorganisms, which include the groups of bacteria and fungi. Decomposers feed and break down dead organisms. As they do this, they release carbon dioxide back into the atmosphere because they are respiring. Decomposers also release mineral ions back into the soil for new plant growth. The job of decomposers is really important. If we didn't have them, all of the materials would be locked inside dead organisms, and eventually no new life would be able to form. Now let's look at the carbon cycle. Pause the video, take a look at the pictures, and see how much of it you can already describe. Then press play to learn more about it. We'll start off with feeding, which is just when one organism takes in the organic material of another, which contains carbon. All living things will eventually die. Plants and animals do this, but of course plants don't have skeletons. When an organism is dead, if it's left in the right conditions for millions of years, it will fossilize into coal, oil, or gas. The alternative is that it will be broken down in the decay process, which we just learnt is done by decomposers. As they do this, the decomposers release carbon dioxide back into the atmosphere. This is just showing that. And some of the carbon dioxide may dissolve into the waters and oceans of our Earth. Here's a little dot and cross diagram to test your links in chemistry. Okay, so going back to fossilization, we can burn the fossil fuels in the process of combustion, and this releases energy, but also carbon dioxide. All living things carry out the process of respiration to release energy for their cells, but it also releases carbon dioxide into the atmosphere when it's aerobic respiration. Finally, the process of photosynthesis is a big process that removes carbon dioxide from the atmosphere. This is really important, as you'll soon learn in the climate change video. You'll have learnt more about photosynthesis in the bioenergetics topic. 8. Biodiversity and land use. Biodiversity is the variety of all the different species of organisms on Earth, or within an ecosystem. The rainforest has a really high biodiversity because there are lots of different species there, while the desert has a much lower biodiversity as less species live there. The future of the human species on Earth relies on us maintaining a good level of biodiversity. Let's recall the definition for a stable community. This is one in which there's a fairly constant population size. But why? Well, having a higher level of biodiversity means that species are less dependent on just one other single species for their survival, like for shelter or food. For example, monkeys might live in trees. That's their habitat. But let's say a tree gets a disease and that species is wiped out. Well, if it's a high level of biodiversity, the monkeys can just find other trees to live in, as those will be there too. Let's say there's a species of bird in a desert that relies on a scorpion. When the scorpion dies, the birds will also die, as that's their only source of food, as there's a very low biodiversity. So biodiversity is important for species survival. But humans, unfortunately, have been doing activities that reduce biodiversity, and it's only recently that we've recognized this and tried to stop it. Let's look at three main ways in which humans have reduced biodiversity. First of all is building. We clear huge amounts of land, of animals and plants to make way for our buildings. Secondly is quarrying. This uses explosives to blow up parts of the earth to get the materials that we want from it. Of course, this is really destructive to vegetation and animals living there. And thirdly is landfill. These horrible mounds of rubbish unfortunately ruin habitats and can even be toxic. 9. Pollution and waste management. Humans have lived on this planet for thousands of years, but in relatively small numbers. In the early 1800s, our population passed the 1 billion mark for the first time. In the last 200 years, it has skyrocketed to over 7 billion people, and the United Nations predicts there will be nearly 10 billion people by 2050. In addition to this huge number of people, our standard of living is definitely increasing. This means that we are using more and more resources and producing more and more waste. And if this waste isn't handled properly, it can cause serious pollution, which is reducing biodiversity by killing animals and plants. There are 3 types of pollution that you need to know about. The first is land pollution. In the previous video, you saw that landfill sites destroy natural habitats, and the household waste can spread toxic chemicals from the waste into the soil. Pesticides and herbicides are one example. They can get into food chains when plants covered in the toxic chemical are eaten. The further up the chain you go, the more toxic chemical accumulates, which can end up poisoning top predators. The second type of pollution is air pollution. When we burn things, the smoke that comes off it contains tiny solid particles that can hang around in our atmosphere. Humans and animals breathe them in, causing damage to the lungs. Acidic gases like sulfur dioxide are also formed when fossil fuels are burned. This then dissolves into rainwater, forming acid rain, which can kill animals and plants in lakes. And thirdly, we've got water pollution. This includes pesticides, herbicides, sewage, and fertilizer. All of these can get into our waterways, but the last 2 contain nitrates. When they're washed into the water, it can cause algae to grow very rapidly, but it will then die. And when this happens, microorganisms break it down, but they use lots of oxygen out of the water to respire. And this low oxygen level means that aquatic organisms like fish die. One tip to remember these 3 types of pollution is LAW, land, air, water. And the link is that perhaps we need more laws to protect our environment from this pollution. If we don't, biodiversity will continue to drop and drop. 10. Deforestation and peat bog destruction. Peat bogs take thousands of years to form from dead plants. They're very acidic, and so they provide a unique ecosystem for lots of organisms that depend on it. For example, Venus flytraps. Peat can be taken from the peat bogs and used as fuel, but when it burns, it releases carbon dioxide. It can also be used as garden compost by leaving it to decay, but as it decays, it also releases carbon dioxide. Destroying peat bog reduces biodiversity as species lose this unique habitat. Deforestation is also reducing biodiversity, which is when land is cleared of its trees. This is happening particularly in tropical areas like rainforests. It's cleared for a few different reasons. First of all, to make room for cattle, and also for rice fields. And the third reason is to make room for crops that are used to make biofuel. Species like orangutans are now facing extinction as their habitats are taken from them. There's another impact, and it's to do with carbon dioxide. Pause the video and see if you can make the link between deforestation and carbon dioxide. Okay, well, deforestation increases carbon dioxide levels. Normally, trees absorb carbon dioxide during photosynthesis, but less trees mean there's less absorption of carbon dioxide during this process. If the cut down trees are burned, they'll also release more carbon dioxide into the air. You'll learn more about the impact of this gas in the next video.
11. Global warming.
Global warming is the increase of the Earth's average temperature. It's caused by two gases, carbon dioxide and methane. In the previous video, we learned that the burning and decay of peat releases carbon dioxide, and we saw that deforestation also causes an increase in the levels of this gas. Burning fossil fuels is another huge contributor to these increasing levels. But methane is also on the rise. Cattle release methane as they digest grass, and rice fields release it as they grow. Both carbon dioxide and methane cause global warming by creating a greenhouse effect. This is when solar radiation from the sun is prevented from escaping our atmosphere by the blanket of greenhouse gases. Instead, it gets re-radiated back to the Earth, increasing the surface temperature. It's important to know the difference between global warming and climate change. As I mentioned at the start, global warming is the Earth's average temperature, but climate change is more severe weather patterns. If you're given one particular phrase in an exam question, you should use that same phrase in your answer. Global warming and climate change will have biological consequences. There are 4 you need to know. First of all is a loss of habitat. When the sea levels rise, it will cause flooding and coastal habitats will disappear. This will cause a change in distribution of organisms as well. For example, grizzly bears will be able to spread further north as it gets warmer, but polar bears' range will shrink as they lose the ice caps. Global warming will also cause changes in migration patterns. Many species of birds, insects, and some mammals move around the world with the seasons, but this may change as the climate changes. And finally, it will cause reduced biodiversity. Climate change will cause the extinction of many species, as they are unable to survive in a changing environment. I have one final thing to mention that is slightly hidden in the specification. That is that there is scientific consensus, which means agreement, that global warming is happening and climate change will occur as a result. This is based on systematic reviews of thousands of peer-reviewed publications. To sum it up, there's a huge amount of evidence that global warming is happening.
12. Maintaining biodiversity.
Scientists and concerned citizens have put in place programs to reduce the negative effects of humans on ecosystems and biodiversity. These include breeding programs for endangered species, like this golden lion tamarin, which can be pretty hard as some species take a long time to reproduce, and it's important to prevent inbreeding. Even if it is successful, often the species' natural habitat has been destroyed, so there's nowhere to safely release them. Another method is the protection and regeneration of rare habitats. This will secure a home for the species that live there. These habitats include wetlands and coral reef. Thirdly, when farmers grow only one type of crop in a field, they've been encouraged to reintroduce hedgerows, which are these bushes and shrubs that separate fields, and field margins, which are unfarmed areas around the crop. This will allow wildflowers and other species like insects and birds to live there. And this is the same for hedgerows. They provide a home for lots of countryside animals. Okay, there's two more methods you need to know. This is the reduction of deforestation and carbon dioxide emissions. But this is only by some governments. Unfortunately, it's not being done worldwide, and some countries refuse to acknowledge the level of threat that we're facing from climate change. And finally, 5, recycling resources rather than dumping waste in landfill. This would help to reduce the toxic waste entering our soils and waterways and reduce the consumption of new materials. Okay, well, this topic is likely to come up as a 6-mark question, so it's important that you do learn all of these methods. A good idea to help you remember them would be to come up with a mnemonic. We'll come up with one now. So let's just recap what each letter stands for. B is for breeding endangered species. PH is protection of habitats. FMH is field margins and hedgerows. DCO is reducing deforestation and carbon dioxide emissions. And R is for recycling. So these are our 5 methods of maintaining biodiversity. Now we'll go through and make a mnemonic, which is a silly sentence to help you remember these letters. You can learn more about this in my mnemonic video, but here's one that I've come up with. Big pandas have frequent madness. However, doctors can only record. So I'm imagining a grumpy panda and a doctor standing nearby looking at them and recording their observations. Time to check. So, can you remember what the mnemonic is? Pause or just say it out loud, and maybe this silly illustration will help. Here it is. Big pandas have frequent madness, however doctors can only record. Now pause the video and see if you can say the 5 methods of maintaining biodiversity using these letters. Here they are. Let me know, how many did you get right? You can leave a comment below saying how many you got out of 5. Let me know if you've got any questions in the comments below. And if you found this useful, please use that red button to subscribe and recommend me to your friends. Remember, sharing is caring. You can also support me by buying me a virtual coffee on ko-fi.com. The link's below. And thank you again so much for watching my videos.